Hi, welcome to Neuroverse,
a podcast hosted by Carolina
and Clara, where we discuss all matters from neuroscience to philosophy and beyond.
Today's episode is on challenges with collaborative neuroscience and how generative AI language models are impacting the world. To help us with this topic, in today's episode we are joined with Jai Bhagat. Jai is from the US and got his bachelor's in neuroscience from Boston University and then worked as a technical associate in Matt Wilson's lab at MIT, working with Carmen Varela researching the role of the thalamus in spatial navigation. He also created deep and unsupervised learning tools for neural event detection and classification. After this, Jai came to UCL and joined Matteo Carandini and Kenneth Harris's lab as a software developer and developed a complete experiment control software toolbox called Rigbox, which we'll discuss a bit later on, and an analysis pipeline for the International Brain Lab to analyze neuropixel recording data. He is currently a PhD student at the Sainsbury Wellcome Centre in Tiago, Branco and Mrsic-Flogel's lab, performing experiments involving continuous recordings of freely moving rodents during decision-making tasks in naturalistic environments. Welcome to the podcast, Jai.
Yeah, thanks for having me. You guys have such official podcast voices. Sounds great.
Nice.
Like, I feel like it sounds like I'm listening to a podcast while I'm here in person. Yeah, it's nice.
We're really happy to have you on, Jai. So in today's episode, I'm really looking forward to hitting a few points from the more concrete day-to-day reality of collaborative science, namely focusing on the International Brain Lab stuff, but also more philosophical discussions regarding generative AI and how they impact the world. So to start off with, I was hoping you could explain a bit what your research is at SWC, what's your research question, and what got you into the topic?
Yeah, so at SWC, I'm right now working with Tom and Tiago, and then a small group of other researchers, and we're doing, it's basically this collaborative project to build a platform to explore naturalistic behavior. So the idea was we wanted to build like the software and hardware components of this platform to do like long-term continuous recording. So we're talking like weeks or even months at a time, continuously 24/7. So that was like the first goal of this project, which we call Project Eon, for like long indefinite periods of time is where the namesake comes from. So yeah, within that it was like, okay, now we've built this platform, or we're thinking about building this platform to explore a large array of behaviors. Like, which behaviors would you be particularly interested in? And the way it works is different PIs at SWC like propose projects within this platform. And Jeff Ehrlich and Antoine have been doing a lot of stuff with like social behavior. And they proposed what I thought were some really interesting social experiments that I think it would be cool to like kind of probe in this long-term continuous setup. So stuff like, can we figure out like the neural basis of cooperation in a foraging setting? Can we teach mice to cooperate just first in general? Do they like switch between cooperation and competition depending on how an environment changes? So all these kind of questions relating to like social learning within this environment is, I guess, my more specific PhD project. I'd call it social behavior and foraging. And then the general stuff is like just the building of the platform.
So when we're talking about naturalistic behaviors, what do you exactly mean? Because of course it seems obvious that it's important to understand more naturalistic behaviors in a research setting where we usually have like quite artificial experimental setups for mice where they're like fixed in one location, they can't really move, they're alone, they're in this like fake environment. But how naturalistic can we make it if you're still in a lab,
Yeah, so I think it's a great question. Yeah, exactly as you put it, there's kind of like these two domains of neuroscience. I'll generally classify them as two domains. One is like kind of like very task-specific experiments, which I think are very useful, right? But they impose these artificial constraints on animals, right? So in a lot of cases, we'll head fix mice or rats, or we'll only have them do one specific thing in a very constrained environment, like with visual tasks, you present some visual stimuli and they have to respond to that visual stimuli. But it's often things that they wouldn't do in their kind of ethological repertoire, if you want to call it that. So for us, it's like we wanted to explore something that we didn't have to train, first of all, so just like put them in and let them do whatever. And we realized that, all animals need to forage, right? In the wild, they need food to survive. So this is what we mean by, I guess, naturalistic, is everything for us centers around foraging first. In our setup, in our environment, we have like this big circular arena where there's like patches embedded into the floor, and they spin a little wheel in order to get food. So the spinning is like a simulation of a digging movement. And we don't have to teach this at all. Like if you put mice in this arena, they just start, and there's like these patches, they find patches, they realize there's like a wheel there, and they start playing with it immediately. So it's like there's no teaching, and as long as you like, you know, are delivering reward after they spin X amount of distance, after they forage X amount of distance, then there's no teaching that you have to do at all. So in that sense, it's naturalistic, so we don't have to teach anything, but then we can still explore, like, without imposing any constraints on the animals behaviorally, we can explore what else they do in the meantime. And then you can add different stuff to that environment. So if you want to add noises or visual stimuli, you can add that and then see how that changes in relation to the foraging behavior.
Yeah, that makes a lot of sense. And then what type of data do you acquire? Yeah,
so right now we acquire like video and auditory data. So we have ultrasonic microphones and we have a bunch of cameras around the arena. We're starting EFIS soon, which will be fun. So we're going to also be using the NeuroPixels too. And that's going to be a huge challenge, just recording 24/7 with EFIS data. Yeah.
For what, you said weeks to months?
Yeah, exactly. So we haven't decided exactly. I think most likely we're going to take an approach where we do like X hours on, X hours off per day.
Yeah.
Because I think it would be too much to do full 24/7. But yeah, it'll be fun to play with, I think.
And what specifically would you be recording? Like what brain areas, what kind of questions are you addressing?
Yeah, so for me in particular, so there's going to be a bunch of projects in this whole platform, but for me in particular, I think there's a bunch of regions kind of implicated in social cognition in rodents and non-human primates, and those are like different cortical regions like OFC, PFC, midbrain regions like striatum, VTA, nucleus accumbens. So yeah, I was talking to people like Dario and Jeff and Anne. We did like a brief literature review and we're like, where can we stick a probe to get as many regions as we can of these like cortical midbrain regions? So we'll try and target like 5 to 10 regions at once and then kind of see. I think the first questions will be regarding like how they share information with each other. So are they learning from each other, kind of like observational learning? And then we'll see like which brain regions correlate with that behavior, and then we'll try and do causal manipulations, like kind of like the classic systems circuit neuroscience approach.
That's really interesting. Yeah, I remember when I first joined in SWC, there was this big debate regarding overtrained mice in science and whether it is representative. So I really appreciate this focus on having more ethologically relevant environments, and then potentially that would be way more reproducible in humans and translational, because we're no longer forcing them to do these unnatural tasks, to distinguish between stimuli that they wouldn't encounter in the wild, etc.
Yeah, there's something nice about like exploring what something was made to do rather than like forcing it to do something it necessarily wasn't made to do. And obviously there's a trade-off, right? There's a lot of constraints or conditions we might not be able to like impose, but I think you need both approaches in science generally, kind of like hypothesis driven versus more data driven.
Do you see a lot of variation between mice with this approach? Because I know, like I was thinking about with the classic training paradigms where it takes like, you know, weeks to train mice. Some of them just learn immediately, some of them take really long. So do you think like with more ethological behavior, would there be more or less variation?
Yeah, so I think we've noticed for the initial learning, it all happens we've done, you know, tens of mice now, maybe like 20, 30 total so far. And the initial learning, just like, you know, that they have to forage via this kind of digging motion to get food, that happens within a few hours always. But then the differences in learning come out when we play with different parameters of the environment. So like if we have 3 patches and we make one patch easier to get food from than the others, then there's definitely variations in that. You know, some mice will learn that. There can be variations like 3-4x in terms of where like one mice will learn it in 1 hour, the other might learn it in 4 or 5 hours. So we definitely see variations that way. But it's nice that the core behavior itself, there's a little variation, right? It doesn't.
Natural.
Yeah, exactly.
So you mentioned that this project Eon, it's a collaborative effort with a lot of people. Could you speak a little bit about the intricacies of making it work? So I was there for your upgrade, and you talked about like the data architecture and data analysis, and these are things that I think sometimes experimentalists just take for granted, like, oh, and they'll figure out later. So what is it like to create a data architecture that is efficient and to create this pipeline?
Yeah, so I can start with a little history if you want to, like how everything kind of evolved, even pre-me. So what I've heard is that Tom Mrsic-Flogel, Tiago Branco, and Troy Marguerite kind of had this idea of wanting to do, and I guess Dario Campagna, who's a postdoc with Tiago, the 3 or 4 of them had originally had this idea of wanting to do this collaborative project within the building. So it started off with, we should be doing collaborative science, and like what is a good way to like get kind of the building involved in this. And Tiago, his lab, naturally does a lot of naturalistic behavior in general. So they kind of started with that, and they're like, okay, if we start with this naturalistic kind of approach to doing behavior and make it collaborative, what is something we could do? And I think the four of them were like, let's do long-term continuous. This isn't something that's really studied in neuroscience, but it fits in naturally with naturalistic behavior. Like, you want to study this behavior over like weeks or months at a time. So I think kind of those 3 or 4 people were like, let's start this big project. And then Gonçalo Lopes, who developed Bonsai, which is a popular software program language that neuroscientists use, they kind of recruited him to use Bonsai as the core of data acquisition, like experiment data acquisition during these long-term continuous recordings. So this is kind of how the project started. It was like, let's do long-term natural continuous behavior. Let's form this big group to do it. Let's get Gonçalo to head like the kind of experiment control part of things. And then I knew Gonçalo a little bit, and this kind of ties into Rigbox, which we can get into, because we had worked on similar things. Rigbox is kind of a similar, what I think is a worse version of Bonsai that's MATLAB based. So we had known each other from the software side of things from before. At the same time he was telling me about this, I was thinking about what I wanted to do for a PhD, and Tom and Tiago were like, oh, this could be your PhD, and they really tried to get me into it. So this is kind of how the project formed initially. And then we were like, so once I was in, I would say, it was kind of like the 6 of us initially, maybe like me, Dario, Gonçalo, and then Troy, Tiago, and Tom. And then we started thinking about like these different subgroups that you mentioned in your question, Carolina. So it's like, now we have this big project, but like obviously there's a lot to do, so how should we kind of organize ourselves? And actually following from the IBL kind of structure, because Tom's lab is also part of the IBL, we said let's kind of separate ourselves into groups that we think make sense to do, where each group kind of focuses on a different thing. And kind of the main groups that came out of this were mechatronics, which focuses on like the mechanical and electronics components of actually building the environment. Task control, or what we call experiment control, which is like what I mentioned Gonçalo does, like the software for actually running experiments and acquiring data. Then data architecture, which is, I guess, like how to handle the data, how to store the data, how to transport the data across different devices. And then data analysis. These 4, I guess the mechatronics, experiment control, data architecture, data analysis were kind of the first 4 initial groups to form. And then we kind of had this extra group that came after that, which is what we call experiment protocols, which is where we actually discuss which experiments we're going to run in this arena, or this environment. So yeah, like as I said, the initial thing was create an environment for doing naturalistic behavior, and now once we kind of like did that, we're like, okay, what specific experiments can we run in this setup?
That's really interesting. I remember in your talk you talked about how to make sure that the data is accurate. I don't know the details, but you spoke about introducing these elements that then you could verify whether or not the data was getting translated correctly.
Yeah, in all experiments, even in acute experiments, but definitely for long-term continuous experiments, you want to have what we call like online quality control checks on the data, so like online QC. So that's common examples of that, of where things can go wrong, or like cameras dropping frames. So occasionally you'll like lose frames, and you want to know exactly when that occurred, if you should do something to fix it, how you should fix it. With microphones, similarly, you can get like weird noise, or also just lose connections. So similar to the way you can get drop frames in cameras, you can get dropped samples from microphone. So you just want to have these checks built into place. Then also like we record the actual wheel that the mice are spinning to get food. So we want to know, because we trigger food based on how much distance they spin on the wheel. So we do like if they spin, let's say 100 centimeters, something deliver 2 pellets, stuff like that. So we need to know if any of these components break. So we're like recording all of these signals continuously in real time, but also doing these like checks on these signals to make sure that we get alerts in case anything breaks.
Just out of curiosity, when the mice are doing these experiments, are there any humans around, or do you just leave them?
Yeah, we just leave them. It's great.
Okay, yeah,
It's
great.
amazing.
So it's like literally in the ideal world, it's like we put them in, we leave them do their thing for like weeks at a time, and then come back. Some complications that get in the way of that are cleaning. We're kind of settling on this approach where like maybe twice a week we'll go in there and clean up the environment so that it's just not full of like poop and pee. But and also like right now we're doing a lot of what I call like troubleshooting experiments, where we're still kind of like playing with environment properties and stuff. So those sessions are shorter, like less than 1 day. But we've already done like left one animal in there for over 2 weeks, or just under 2 weeks. I think 13 days is the longest experiment we've done now. But yeah, so that'll be the plan, is like put them in, leave them, go in a couple times to clean, but yeah, that's it.
Nice.
Yeah.
And so this is a very collaborative project, but you've also done other collaborative projects. You worked in the IBL, the International Brain Laboratory. Could you tell us a little bit about your role in that project?
Yeah, so it's fun. I came over to Kenneth and Matteo Carandini's labs initially to work primarily on the software I just mentioned, Rigbox, for running experiments in their labs. But they did a lot of collaborative stuff, and I'd say this is kind of like, I guess, my first introduction to collaborative science, because I hadn't done that much of it at Boston University and MIT before. And so yeah, Kenneth and Matteo are very collaborative, and two big projects they worked on were NeuroPixels projects and the International Brain Lab. And the International Brain Lab, so in general it's like this, it's this group of I think around 20 labs, 9 experimental labs. And the whole idea was, let's get this huge data set of these mice doing one particular task and record from all over the brain while these mice are doing, so get like a full brain-wide map of electrophysiological data while they're doing this one task. You get like a brain-wide map of the mice doing this one task.
Is that with NeuroPixels?
Yeah, that was in NeuroPixels, yeah. And so my role in that was kind of secondary. So I was spending basically like a few 5 to 10 hours a week maybe, doing stuff like thinking about how to analyze the EFIS data. I worked with really cool people, Olivia Winter, Cyril Brisson. So it was really fun. I got to work with what I consider professional software developers. So I got to just like learn a lot from them. But yeah, I was doing stuff like thinking about how to organize IBL-lib, which is their main Python package for accessing the data, acquiring the data, analyzing the data. So when you interface with IBL data, if you're an IBL researcher, you use IBL-lib. So a lot of it was kind of just like listening to them, telling me what to do, looking at things. So I'd like code up stuff and they would like refactor my code to make it better.
Oh, nice.
So for me it was like a lot of learning.
Yeah.
And it was a lot of fun. And I focused, I think, a lot on the reproducible EFIS part of it initially. So one big question was, how can we get reproducible behavior across all these labs? But also how can we make sure that the EFIS is consistent across all these labs? So you want to have everyone kind of doing the same initial quality control checks when they put the probes in, when they're recording. You want to make sure that like everything looks the same across labs that are spread across, Europe.
Yeah.
And the US. Yeah, so it was kind of just like talking to researchers, thinking about how to code up these pipelines within this IBL-lib package.
Yeah, it's amazing how all of the researchers have this protocol that they follow, and they did successfully make the EFIS data reproducible. And I think it's the number one priority in science, or it should be. I think it's amazing that there is this effort to make it more collaborative, more reproducible, with more and more collaborative groups coming along. What do you think are the main advantages and disadvantages of having such large collaborative groups?
Yeah, I think obviously the advantages are huge. So it's funny, we just had this conversation a little bit with our friend and colleague, Nate Miska, because Nate works in the IBL also. And I think the advantages are kind of obvious. I think we're past this point, at least in systems neuroscience, there's a lot of early research looking at individual cells and saying like these hypotheses that these cells code for a specific thing. So coming up with concepts like place cells, grid cells, face cells. But now we're at the point where we're like, okay, obviously it's not just individual cells that are doing stuff in the brain, but it's like how they communicate with each other. So really looking at populations of cells. And when you start having recordings with thousands or potentially tens of thousands of neurons with imaging tools, you really need to think about how you're acquiring that data, how you're analyzing that data. And I think the data has grown to a point where it's kind of infeasible for one person, one PhD student, one postdoc, one RA to have a project by themselves if you're really trying to answer like a big kind of general question. So I think that's the immediate obvious pro of collaborative approaches is you get multiple people working on these huge data sets. You can get specialization, so you get somebody collecting, somebody analyzing, and they'll often work together. So it's not one person who's only collecting, but they're primarily in charge of collecting data, and then one person who's not only analyzing, but analyzing some, but then also seeing how the experiment's carried out. So you get like specialization, but also collaboration within these specialization groups. And I think, you kind of need this kind of like division of labor when you have these very big projects.
You also mentioned that you worked with NeuroPixels in Matteo and Carandini's lab.
Yeah.
So what was your role on the pipeline?
Yeah, so the NeuroPixels project was this huge project, which it's funny now, the NeuroPixels I think are like the predominant de facto form of doing EFIS in systems neuroscience. And so it started, there's a postdoc, Nick Steinmans, who was kind of the first author of both the NeuroPixels 1 and 2 papers. And he started, should I give the history of like how I came to that briefly? Yeah. So Nick, so it was really funny. So Nick had been doing all these experiments probably like 2016, 2017 that were the initial test NeuroPixel probes. And I was at MIT at the time, and I was working on spike sorting, which is basically when you record EFIS signals from extracellularly from the brain, you get like a bunch of spikes, And the whole goal is like figuring out which spike belonged to which source neuron just from the spikes themselves. And I was working with tetra data at the time, so like these wires that would consist of 4 channels grouped together, and so like one tetra drive array, you'd maybe have like 100 channels total, maybe like 128 channels is the drives we worked with. And then I heard about these neuropixel probes from a talk Kenneth came and gave detailing neuropixel data. And I was like, oh wait, these things have like 380 channels. This is crazy. I wonder like how they're thinking about spike sorting this stuff. So then I got in touch with Kenneth, and I was like, oh, the stuff that your lab is doing seems really cool, and it'd be cool to analyze this data. Can I play with it? Kenneth and Nick were nice enough to give me data to play with.
Nice.
So I worked on spike sorting for that, but then of course, smarter people develop better spike sorting algorithms, but it was fun to at least like play with that data. And so that was kind of my first introduction to the Cortex lab. Matteo and Kenneth was through the NeuroPixels project. And then I came over through communication with them, and I was working on a separate project, Rigbox. But then they were like, okay, we want to make these changes to the NeuroPixels probes, and they talked about creating this NeuroPixels 2 paper. And so my role in that was pretty small, but I was working with Anna Lebedeva, a PhD student now who just finished in Cortex lab, and Nick a little bit to analyze. So they had similarly to the IBL, there's about 4 or 5 experimental labs that were playing with the NeuroPixel 2 probes. And one big thing we wanted to do was track units over time. So we wanted to say, if you have a chronic implant, how can you say that the spikes from this unit, are the same spikes that you'll get on 2 or 3 days down the line? Like it's the same unit, and you want to track that unit over time. So the way we did that was with visual responses. So I was looking at V1 data from a few different labs, including Anna's data. So you basically present a visual stimulus, and you get spikes that respond to that visual stimulus. And you know that that's probably 1 unit. So you want to see the degradation of those spikes over time, because you'll probably lose that unit over a few days. So you kind of want to see after how many days, when you present this visual stimulus, do you stop getting these spikes? So how long can you track this unit for, basically?
Why would you lose the signal from?
Yeah, so there's a couple reasons you can lose cells chronically over time. I think the 2 main ones are just cell death. So you'll have tissue death from the implant. Basically what happens is when you insert a probe, you'll get cell death, and that cell death can either kind of like spread, or there's like, I think the tissue tries to correct itself. It literally forms what you can think of like as scabs on the skin. So it makes it harder. You like kind of lose the area that you can record from, because now electrical signals aren't penetrating through those like neural scabs as well as they would as just clear tissue. So that's one reason. The other reason is that you get commonly referred to as neural drift. So like the probe will drift slightly in relation to the brain, or you can think of the brain as drifting slightly in relation to the probe. So one cell that you're recording from, it can slightly drift away from the probe over time. And so then you'll lose the ability again to record from that cell, because it's physically getting further away in space.
I want to do a quick change into the topic, and I want to talk about generative AI and language models, because I know it's a big interest of yours. And so for our more experimental audience, could you explain a bit what language models are, what makes them so powerful? And I know you have a lot of opinions on them, so let us know. So as a caveat, a precursor, I'm not like studied AI by trade or anything, but I think I grew really interested in generative AI probably a little less than a year ago now. I think it was kind of at the time GitHub Copilot and Midjourney. So GitHub Copilot it's based on this language model called Codex, but it kind of like auto-completes code for you as you write. So if you're writing code, it will predict what you're going to write, and oftentimes it's very powerful. So I use this as a plugin into GitHub, and I was like, wait, this thing can read my mind, and it like saved me so much time just coding. And then I was also playing with tools like Midjourney, which it uses text-to-image generation. So you type in prompts, and it creates images from that. And there's a bunch of tools to do this now. When I first started playing with these tools, I guess a little less than a year ago, I was like, wow, these are incredibly powerful. So then I was like, kind of as a side thing, was like, okay, I want to learn more about these. And then of course ChatGPT dropped in say like November last year, I think. And that was just like a huge game-changing thing for me, just in terms of work initially. So I was using like Copilot already for code, but then I started using ChatGPT a lot for like honestly everything. So I'd do stuff like, okay, we're recording. We did it a lot for Project Eons. I'd ask questions like, okay, we're recording from all these data streams. These are potential ways that we could save the data, and these are potential ways that we could transport data. And I'd like
put my ideas into ChatGPT and be like, what do you think is the best approach to do this? And it'd come up with stuff that was like pretty reasonable, and then I'd ask experts and they're like, oh yeah, you should do what it says. And then I'd also just use it as review, so I'd like, post in snippets of papers and be like, can you explain this to me in simpler terms? All the common use cases that people use it for.
Yeah.
So I was using it for a lot of things. And I guess that was my introduction to Gen AI. And then from there, things have grown more and more powerful, especially in the recent months. I think after GPT-4 it was released in February. And I just realized that there's all these applications, I think primarily for, right now it's very easy to get into it if you're doing anything the sciences or data science. And I think one big reason that is probably the data sets that GPT 3.5 and 4 were trained on were a lot of academic papers and a lot of code. Because these are considered high quality data sets in natural language processing. I think every researcher should be using these tools as much as possible, because you can plan with these tools, you can ask it about your research program, they can usually give you good insights, you can code with these tools, you can learn a lot from them. There's all these kind of, people are now creating companies revolved around using language models for educations and like personal assistants and tutors. Yeah, generative AI in general, but language models maybe more in particular are just going to revolutionize the world. And yeah, I mean, Caroline, I know you've already been playing with ChatGPT a lot, so you probably have similar opinions.
I mean, I've been trying to use Sleep, which is this estimating deep learning software, and I don't know any coding, and ChatGPT basically helped me do most of my analysis. So I do have to give it a lot of credit. I remember you were having this discussion with Joe about how, yes, they're really powerful, but also they are trained on these data sets, and they also are trained on people asking them questions and stuff. So, and you had this conversation about like garbage in, garbage out, and like what is the appropriate like data sets for it to be trained on and stuff. Could you expand on that? So
there's this whole, I guess the way, from my understanding, the way kind of language models developed was initially, the whole concept is based on this network architecture called a transformer. And the way, at a high level, the way a transformer works is it takes input, it processes all of that input at the same time. And what it does is then it starts generating tokens from that input. And a token you can think of as a subword, so just like parts of a word. And it keeps generating these parts of a word until it reaches what is called an end token. So it has some notion, given a certain context, your prompt, of what an end token will be like for that prompt. So, and the way it produces these tokens is it has this probabilistic map of all the tokens in a certain language. So with English, there might be, 400, 500 tokens that it produces. And given a certain prompt, some of those tokens will be end tokens. And it just has a probability of it's like a softmax over all of these tokens. And it keeps picking these tokens one at a time until it reaches an end token. And that's roughly how a transformer works. And then so it's like if you just input stuff, it'll start outputting stuff until it reaches an end token. But it's trained on stuff on the internet. So like common things that you could train it on are like a forum or a list. But then how you divide this up into what is like the training data versus the evaluation data, it can be arbitrary, it can be up to the person. So for example, if you are giving it a list of questions, what you might do is, one way to split this data up is say there's 10 questions you're giving it. You might split it up. The first 5 questions you train on, it gives some output, and you want to assess its output based on the last 5 questions. So that you would want it to come up with the last 5 questions as its output. It could be like a list of like what are the capitals of certain countries. So what is the capital of France, Spain, England, and then like that's what it'd be trained on, but you want it to output then what is the capital of the US, what is the capital of Mexico, whatever. Right? So that's one way you could train it, but the other way you could train it is give it all the list of questions and then have it come up with answers instead. And people found that's typically more useful, right? Rather than if you input a question, you don't want it to come up with another question. Ideally, it would come up with an answer. That seems like a useful way for most people to interact with this thing. So this big step was called instruction tuning. So get it to respond to instructions. And then I think this happened around like GPT-3 or right after GPT-3. And this was a big foundational moment for language models. Let's focus on instruction tuning because that's what people find useful. You can have kind of useful information that you input or bad information that you input. So it could be, if you have bad information, that's this term garbage in. If you feed in garbage in, you'll get garbage out. So in my toy example, like the garbage in could be if you just feed in the questions and you're evaluating it on creating its own questions in response to those questions, that could be considered a garbage data set for most people, right? Because they want answers to the questions rather than having it come up with its own questions. Okay, so yeah, better data in the case you feed it all the questions and you evaluate it, the answers it comes up with to those questions. And that would be, for most applications, non-garbage data. So you get like, I don't know what the opposite of garbage in, garbage out is, but like good in, good out.
Yeah.
Yeah.
So even if you get good in, good out, does the out surely still depends on the in? So like, would it be beneficial to have different generative AI tools that are trained on different types of data, like academic papers and code, like you said, ChatGPT was, and like another one for something else, something lower level?
Yeah, I think that's totally, so I think what's incredible, why ChatGPT is so good, I think, is we don't know what they trained on. I think they basically trained on like the whole internet, and they probably trained like anything that's publicly available up until 2021, and they must have used the same example millions of times, right? So they kept training on like thousands of papers, whereas tens of thousands of forums, tens of thousands like code repositories, and they kept training on those same kind of individual training items millions of times probably. So that's what makes it such a good general tool. There's this big open source movement now to make smaller language models like you suggest, that are focused on specific things. And I think this is something I'm really interested in. I can still use like tools like ChatGPT for my own data, but I have to spend a lot of time explaining to it what my data looks like before it will be like, okay, here's reasonable analysis to do.
Yeah.
But it would be great if I could just feed in the whole context of my experiment. Like a way that like, okay, now it has the whole context of my experiment, so I don't have to, every time I ask a question, I don't have to keep giving it the context. It's something that has like been trained, been fine-tuned, is the terminology used, on my whole data and like questions that I already care about.
Yeah.
And I think this is becoming popular. People are already doing this, and it's something I want to play with on my own now with smaller open source models. So there's models that are basically derivatives of Meta released this open source model called Llama. That's again a smaller version of ChatGPT, a transformer-based model. And yeah, a lot of people have now like created a lot of the specification or specialities of language models for particular use cases are kind of based on now derivatives of Llama. And I think this is something I'm interested in exploring myself with my own data, because then you could have something that's offline that has full context of your experiment.
Personalized.
Yeah, and I think this is the future of generative AI. I think you'll see like more and more of these smaller models popping up across industries, really.
That's amazing.
Yeah.
Cool. That sounds super exciting.
Yeah.
We're running out of time.
Yeah, sorry.
So no, it's okay. I want to talk about one more thing, because I know you're really passionate about education. So do you want to tell us a little bit about neuronautics and how education plays a crucial role in future neuroscience and science in general? Yeah,
so I think a key aspect of science should be outreach. So if you're an amazing scientist but you don't know how to communicate your results, then you're worthless, right? You're not doing science. Like, it doesn't make any sense, right? Yeah. Like you want to communicate with the general population, but then you also want to, I guess, teach people how to, interpret what you're sharing with them. So it's kind of you communicating with them, but you teaching them as much as possible about what you do. The outreach stuff seemed like a natural, if you're doing science, you should be interested in outreach. It just seemed natural to me. And I think it's really, I get a lot of fun out of like seeing people learn stuff and working with students. I think I get a lot of fun out of it because I also learn a lot from them. Because when you see how different people learn something, it teaches you to think of that concept in a new way. And then you might learn something more, like explore a concept more in depth. So I think, I'm really interested in teaching. I really like tools for neuroscience, as we talked a lot about, like both kind of building software and hardware tools. So I like teaching students, and I primarily work with like secondary school students in this group that we've formed called Neuronauts, which is, it's a summer school and now also a school club for secondary school students in London. And we teach them basically, it kind of centers around tools for doing initial neuroscience research, or even like cognitive science research. So we teach them basics in electronics, in programming, and then we give them basic concepts of how we study systems neuroscience as a whole. And like last summer it was the first annual Neuronauts program, and students did really fun experiments. They would go out into the wild. They went out to Regent's Park and they recorded animals in doing naturalistic behavior. So people were like left food out for squirrels and then saw which food the squirrels were most interested in, and they were recording like video and audio data in real time. We did the same with ducks. We had like a little robotic arm that would deliver pellets to ducks in the water, and we were trying to figure out like which sounds would make them approach the pellets, which sounds would make them go away from the pellets. So I think education in general is a natural kind of progression of science, or a natural component of science. I think we should strive towards like a meritocracy as much as possible. Definitely. And also like equality as much as possible in our societies. So we try to work with like students from like lower socioeconomic areas in London. And it's just a way for them to get exposed to this whole lab setup that they might not have otherwise, like a lot of them might not even consider science as a possibility for a career.
Yeah.
And I think because I feel like I've gotten so lucky in terms of what I've done, and it's like just opened my eyes, like things I would have never considered before, like coming to London just happened randomly because I was working on spike sorting in the first place. So I think if you open people's eyes to a lot of things that they had never even considered before, then that gives them a lot more incentive to do things that they didn't think were possible, and they can realize like, okay, I can do this. Like anyone can do this.
That's so amazing. And I think this is a perfect place to end today's episode. Thank you so much for joining us, Jay.
Thank you so much for joining us. It's been really good.
If you enjoyed today's episode, please feel free to share it with your friends, and check out our website for resources and links and papers.
And be sure to follow us on Twitter and Instagram at neuroverse_pod.
Thank you.
Thanks.